All right, hey everybody. This short video is about experimental design and experimental variables. So as you're going to see in my PLTW, if you haven't yet, there is a little bit of an introduction to experimental design. We'll come back to this more in a future activity, but just want to get the basic steps in order to begin with. First step in the experimental design process is to identify the question or the problem. So this is where, we're making an observation, we're recognizing some sort of relevant issue. We're researching into that observation, seeing if there's possibly something to investigate further, and then writing a research question or a problem, right? So in the scientific method, we often call this the observation and the question step. And then we come to making a prediction, step number 2. And this is where we're going to be writing a testable hypothesis and also identifying the variables. When we write a testable hypothesis, it's important to know what our variables are going to be, especially the variables of import. And we're going to get to those important variables on the next slide. Then comes design an experiment. This is where we're going to define our controls and our control groups, as well as any variables that need to be kept constant or controlled. We're going to list the steps of the experiment out. We're going to do that very diligently and have a detailed procedure, identifying any materials and safety precautions, planning for data collection and analysis. And then we actually come to step number 4, conducting the experiment, which is where we're just going to follow those steps that we wrote out in step number 3, collect our data, make any additional observations that might be relevant, that might be influencing our data. And we also want to make sure we're completing multiple trials. So this is where that repeat step that you might be familiar with from learning the scientific method before comes into play. Finally, we have analyzed the data. This is where we're going to be pulling all of that data and observation together to try to interpret and draw conclusions based on all that data that we see. This is where the importance of making charts and graphs, various visual aids, different ways to represent that data is going to be really important. And then communicating findings. Of course, any sort of experiment is only going to be useful if it is well communicated to other people who might be interested. So we're going to reflect back on that original question. We're going to, do some scientific writing, as well as outline next steps. Where should we go from here? So those 6 steps of experimental design from PLTW. That's what they've got for those 6 steps in order. So identifying the question, making a prediction, design an experiment, conducting an experiment, analyze the data, communicate the findings. Make sure you know those steps in order. All right, next up, experimental variables. So an important part of step number 2 in that making a prediction step when we're writing a testable hypothesis is identifying the variables. The variables are really important. Often we want to write our hypothesis where we have our variables very clearly part of our hypothesis. So you've maybe had to do some hypothesis writing in an if-then format. Often the way we do that is we say, if we make this change to this variable, then we expect this change in another variable. And those are the 2 variables that are most important for our experiment. So the first variable there is called the independent or manipulated variable, the one that we are changing. So whatever variable is being manipulated by a researcher, that's the independent or manipulated variable. So for example, if you were going to conduct an experiment where you were going to test the effectiveness of different fertilizers on plant growth, your independent variable would be those fertilizers, right? That's the thing that you're going to be changing between your different experimental groups. This one gets fertilizer brand A, this one gets fertilizer brand B, this one gets fertilizer brand C, and maybe you have a control group that doesn't get any fertilizer at all, right? So that's that independent variable thing that's different between our different experimental groups. Then you have your dependent or responding variable. So this is the thing that you are expecting to see a difference in, something that is depending on, right? Depending on the independent variable is the thing you expect to respond, right? So when we manipulate this, we see a response here. And so for our fertilizer experiment example, right, we've got plant growth, right? Whether that's height, size of leaves, could be a whole bunch of different variables that you might be looking at specifically, but plant growth would be our dependent variable in that experiment. And then you have controlled or constant variables. These are variables that you need to keep constant and control for in every experimental group, every trial run. So for example, with the fertilizer example, how much sunlight they're getting, how much water they're getting, the temperature of the environment, a whole host of other factors you could probably think of as well. But these are all things that you would want to have the same, kept constant, right? You're controlling for those. There's an announcement. All right. Have a good one, guys. Thank you.